Hello, everyone, and welcome to today's session. And today we'll be discussing the NCERT exercises for the chapter Cell the Unit of Life. Before starting lectures, we want to give you one information that the images, text, and other materials used in the presentations are for education purposes only. We respect the intellectual property rights of the original creators and owners. If you are the owner and have any concerns or issues, please contact us at sathi@itk.ac.in and we shall remove the content immediately. Please note that parts of the content may have been generated using generative AI, which is experimental. What are the objectives of today's session? So, today we will explore and we will understand the basic concepts related to the chapter Cell the Unit of Life by working through the NCERT exercises. So, these exercises they are based on certain concepts. So, we will understand these concepts by discussing the NCERT exercises. Okay, so there are 14 questions. We'll try to discuss all these 14 questions in this session. Let's begin with the first question. The first question is which of the following is not correct? Okay. So you have to select a false statement here. What are the options? First option, Robert Brown discovered the cell. Is it correct statement or is it incorrect? Next is Shachalidan and H1 formulated the cell theory. Is this correct or not correct or incorrect? Next is Virchow explained that cells are formed from pre-existing cells. And option number D is a unicellular organism carries out its life activity within a single cell. So which is incorrect statement? Let's have a look at the answer. Okay, answer is option number A. Robert Brown discovered the cell. This is incorrect statement because Robert Brown he discovered this cell. So cell was first discovered by Robert Hooke. Okay, he studied the cork cells, which are dead cells. Then live cells they were discovered by Leeuwenhoek. Next one, new cells generate from. Okay, what are the options? New cell generate from bacterial fermentation. Option would be regeneration of the old cells. C is from the pre-existing cells and D is abiotic materials. What is the correct answer? Okay, the correct answer is option number C. Okay, correct answer is option number C, pre-existing cells. Okay, so this is according to the cell theory that cell arise from the pre-existing cells. This was given by Rudolf Virchow. Okay, the new cell can arise from only the pre-existing cells. Clear? Now, next one, this is the match the following question. Okay, this is a column based question. You can see two columns are given to you. In first column, what is given to you? The name of the structure, cristae, cisternae, and thylakoids. Okay, and in option in column number 2, the explanation of these terms are given to you. Okay, where these structures are present. So you have to match column 1 with column 2 here. So what would be the correct answer? Okay. So A matches with what? B matches with what? C matches with what? So let's have a look at the answer. So you can see cristae, they are the infoldings in the mitochondria. So C will be 2. Cisternae, they are disc-shaped sacs in the Golgi apparatus. And thylakoid, they are the flat membranous sac in the stroma. In thylakoid, light reaction occurs. And the stack of thylakoid, they are known as the grana. So this will be the correct answer. Okay. So cristae, they are the infoldings in the mitochondria. Cristae are the inner membrane folds of the mitochondria. Okay. These folding increase the surface area for ATP production. Okay. Now, cisternae, they are disc-shaped sacs in the Golgi apparatus. So, cisternae are flattened, membrane-bound sac present in the Golgi apparatus. They are arranged in parallel stacks. Then thylakoid, they are flat membranous sac in the stroma. Stroma is the fluid of the chloroplast. Okay, in thylakoid, l- light reaction occurs because thylakoid consists of the chlorophyll. Next question, which of the following is correct? You have to choose the true statement here. The options, first option is cells of all living organisms have a nucleus. Do all cells have a nucleus? Or are there are some exceptions? Next is both animal and plant cell have a well-defined cell wall. Option number C, in prokaryote, there are no membrane-bound organelles, and D, cells are formed de novo from the abiotic materials. What is the correct answer? Let's have a look at the answers. Option number C is correct. Okay. In prokaryote, there is no membrane-bound organelles. They lack true nucleus also. Instead of nucleus, they have a nucleoid. Okay, so membrane-bound organelles, they are surrounded by a single membrane, not double membrane. Like in eukaryotes, you have organelles. Okay, most of the organelles, they are surrounded by double membranes, but there are some organs which are surrounded by only single membrane. Okay, nucleus, mitochondria, chloroplast, these are some example of such organelles. These membrane-bound organelles are absent from the prokaryote. This is a prokaryotic cell. You can see instead of nucleus, they have a nucleoid. Okay, cell membrane-bound organelles are absent. Next, what is mesosomes in a prokaryotic cell? And you have to mention the function that the mesosome perform. Okay, what are mesosomes? So let's have a look at the answer. See, mesosomes, they are convoluted membranous structure. Okay, or you can say they are the extensions of the cell membrane. Okay, they are present in prokaryotic cells and they are the invaginations of the plasma membrane. Okay, also known as the cell membrane. Its function are as follows. So first of all, these extensions help in the synthesis of the cell wall. It also helps in the replication of the DNA, means duplication of the DNA. They also help in the equal distribution of chromosomes into the daughter cells. Okay, here you can see this is the cell wall and you have the plasma membrane. These are the, you can say, extension or invaginations of the plasma membrane. This is an image showing you bacterial mesosome. Okay. Now next function. This is very important function here. It increases the surface area of the plasma membrane to carry out various enzymatic activities. To carry out various, you can say, enzymatic activity means lot of biochemical reactions. Okay, this is very important function. It helps in secretion processes as well as in bacterial respiration. So they can ask you what structure helps in the bacterial respiration. So that is mesosome. This is very important function. Apart from the other function, this is the very important function. Next, how do neutral solutes move across the plasma membrane? Can the polar molecule also move across in the same way? If not, then how are these transported across the membrane? So plasma membrane, this is made up of phospholipid. It is a phospholipid bilayer. Okay, how will polar molecules then move across the plasma membrane. Okay, polar means water loving. Okay, so what will be the answer? So you can see plasma membrane is the outermost covering of the cell that separates it from the environment. It regulates the movement of substances in and out of the cell. So this is the structure of the plasma membrane. This is the fluid mosaic model. You can see this is a lipid bilayer. This is phospholipid bilayer. Heads are towards the outside, tail is towards the inside. Okay, then you have some proteins also, integral proteins. These are some peripheral proteins. Okay, then you have some sugar molecules also. Okay, you have cholesterol also. So how will neutral and polar molecules travel across the plasma membrane? Okay, so plasma membrane is selectively permeable. It allows only some substances to pass through it. How the neutral solutes they travel across the cell membrane? So simply by passive diffusion. Okay, so they move across the plasma membrane by simple passive diffusion. There is no energy required also. That's why it is a passive process. Okay, so diffusion is the transport of the materials from higher concentration to lower concentration. Okay, then what about the polar molecules? So cell membrane, you know, it is made up of phospholipid bilayers and proteins. So it is difficult to for the polar molecules to pass. So they require some, you can say, assistance. So movement of the polar molecule across the non-polar lipid bilayer requires some assistance in the form of carrier proteins. Carrier proteins, they are the integral proteins. Okay, that have certain affinity for specific solutes. So they facilitate the transport of this molecule across the membrane. This is known as facilitated transport, facilitated diffusion. Facilitated means with the help of certain proteins, okay, carrier proteins. Now, next question, you have to name two cell organelles that are double membrane bound. It's very simple. And what are the characteristics of these two organelles? You have to state the function and you have to draw the diagram also of these two organelles. So let's have a look at the answer. What are the two organs? Mitochondria, chloroplast. Chloroplast is the plastid. Okay, these are two organelles with double membrane. This is the well-labeled diagram of mitochondria. So you will make the outer membrane and the inner membrane. Then between the inner membrane and outer membrane, you have the intermembrane space. You can label the matrix, which is the fluid of the mitochondria. Then crista present, which are the infolding of the inner membrane. You can show some ribosomes as well, which are involved in protein synthesis. Okay, mitochondria has its own DNA, mitochondrial DNA. Next, what are the characteristics of mitochondria? See, mitochondria are double membrane bound structures. Membrane of mitochondria is divided into inner and outer membranes. Okay. Now the outer membrane is very porous. Inner membrane is deeply folded. Okay, these folds are known as cristae. This we have already discussed. Okay. Inner membrane has a very important function. Inner membrane is involved in the electron transport chain also. So it has all the complexes and the proteins which are involved in respiration also. Okay. Cristae increase the surface area inside the cell. They are the site of ATP generation. Membrane of mitochondria contains specific enzymes meant for specific mitochondrial functions. And mitochondria, they are the sites of aerobic respiration. So whenever you talk about cellular respiration, we are talking about mitochondria, which are the site of aerobic respiration. Mitochondria, they have their own DNA and ribosomes. Okay, ribosomes are of 70s type. The next one is chloroplast. This is also a double membrane bound structure. Okay, it has outer and inner membrane. So thylakoids are present within the chloroplast. Stacks of thylakoid, they are known as the grana. Okay, thylakoid consists of chlorophyll molecules. Thylakoids are flattened membrane sacs. So here you can see this is structure of the chloroplast. So you can show these are the thylakoids. Stacks of thylakoids are known as grana. Two thylakoids, they are attached to each other with the help of stroma, lamellae. Okay, you can show the stroma here. Chloroplast has its own DNA. It has its own ribosomes, which are of 70s type. Okay, you can show the intermembrane space here. Okay, this is the grana. This is stroma and lamella. Okay, so we were talking about the structure of the chloroplast. So two grana, the adjacent grana, they are connected with the help of stroma lamellae. Why they are called stroma lamellae? Because they are present in the stroma. Stroma, what is stroma? It is a homogeneous mixture in which grana are embedded. It is the fluid of the chloroplast. Stroma has several enzymes which are involved in Calvin cycle, also known as the C3 cycle. Okay. So these enzymes, they are also used for the synthesis of carbohydrates and proteins. Mitochondria has its own DNA and ribosomes as well. Now, functions of mitochondria. They are the site of cellular respiration. We have seen they provide energy in the form of ATP. They have their own DNA and ribosomes. We already know this. Okay. They are semi autonomous organelles because they can synthesize their own proteins as well. Okay. Some of the proteins, they are synthesized by the nucleus, but they can also synthesize their own proteins as well because they have their own DNA. They are semi autonomous organelles. Okay. They have several enzymes. Okay, which can carry out various biochemical reactions such as fatty acid. You can say a lot of lot of biochemical reactions. Function of chloroplast. So we know the chloroplast consists of the chlorophyll pigment. This chlorophyll capture the light energy and convert it into food. Okay, so it is very important for the process of photosynthesis. The chloroplast, they are present in the plants. They are present in the leaves. They are present in the mesophyll cells of the leaf. Okay, so these chloroplast, within the chloroplast occurs the C3 cycle, also known as the Kelvin cycle. These cycles are involved in the production of carbohydrates and proteins as well. Next question, very straightforward question. What are the characteristics of a prokaryotic cell? Okay, for more information, you can read NCERT section 8.4. So let's have a look at the answer. See, prokaryotic cell, they are unicellular, means they are single-celled organism and they lack membrane-bound organelles. Okay. The prokaryotes, they belong to the kingdom Monera. Kingdom Monera consists of the bacteria, which are prokaryotic cells. So prokaryotes, they are unicellular, they are single celled, they are generally small in size. They have very simple organization. Okay. Instead of nucleus, they have nucleoid. So nucleoid is a region where the DNA is present. Okay. So DNA is negatively charged. So negatively charged DNA is, you can say, bound to positively charged proteins. Okay. So the region where the DNA is present, this is known as the nucleoid. but this nucleotide is bound to positively charged proteins. Okay, which are histone-like proteins. Actually, histones are absent in prokaryotes; they are present in only eukaryotes. Okay, so like in eukaryotes, the DNA is wrapped around the histones. Here also, the DNA which is negatively charged is bound to positively charged proteins, which are histone-like proteins. Prokaryotic cell, what is the size? It varies from 0.5 to 5 millimeter. So milli is 10 raised to minus 3. So this is an image showing you prokaryotic cell. They have 70s ribosomes. They have flagella as well, which provides them motility. Okay, they have nucleoid where the DNA is present. They have the cell wall also. So in bacteria, the cell wall is made up of peptidoglycan, also known as murin. Okay, then they have glycocalyx. Okay, thickened glycocalyx is known as capsule. They have this pilus, which helps in the attachment. Also, they help in the process of sexual reproduction also, or they help in the transfer of, you can say, DNA in the process of conjugation as well. Okay, so they lack true nucleus, means the nucleus is not surrounded by any nuclear membrane, or there is no nuclear envelope, you can say. Okay, they lack true nucleus. So genetic material in prokaryote is naked. It is lying in the cytoplasm. They have single circular chromosome. So eukaryote, they have linear chromosome, but the prokaryote, they have only single circular chromosome. All right. Now, in addition to the genomic DNA, they have small circular double-stranded DNA known as the plasmid. So plasmid is also used as a vector in biotechnology. Okay. So in addition to the genomic DNA, they have this plasmid, which is known as extranuclear DNA also. It is also circular. Okay. Now they have specialized membrane structure known as mesosomes, where which are very important for the bacterial respiration. These mesosomes, they are found by the invagination of the plasma membrane. So this is the image showing you bacterial mesosomes. Okay, so these mesosomes, they help in the synthesis of cell wall also, replication of DNA. They help in equal distribution of chromosomes into daughter cells, we have seen. So this is the cell wall. Beneath the cell wall, this is the plasma membrane, and these are the invaginations of the plasma membrane. These mesosomes also increase the surface area for ATP production, and they are involved in the bacterial respiration as well. If prokaryotes membrane-bound organelles are absent. Now, this is very important. Prokaryotic cells contain three-layered structure. What are these three layers? Outermost is glycocalyx. Okay, middle is the cell wall, and the innermost is the plasma membrane. Okay, so glycocalyx, when it is very thick, it is known as the capsule. When it is quite thin, it is known as the slime layer. Here you can see this is a capsule which is nothing but thickened glycocalyx. So they have glycocalyx, then cell wall, and then plasma membrane. So this glycocalyx protects the cell also, and it helps in the attachment of the cell also to the substrate. Okay. So these structures protect the cell. So they are a protective unit, glycocalyx, cell wall, and plasma membrane. Examples of prokaryotic cells, these are bacteria, obviously. Blue-green algae, which is also known as cyanobacteria. This is also prokaryotic cells, and both of them belong to kingdom Monera. Next question, multicellular organisms have division of flavor explained. So what is happening in the unicellular cell? Single cell is performing all the life processes, such as respiration, excretion, okay, digestion. But in multicellular organisms, okay, like humans and higher animals, different cells perform different functions. Okay, like you have nerve cells which are present in the nervous system. Then you have RBCs. Then you have reproductive cells which are involved in the formation of gametes. Okay, so you have to explain the division of labor in the multicellular organisms. So multicellular organisms, they are made up of millions and trillions of cells, and these cells perform different functions. Okay, so these cells are specialized to perform functions. Okay, and the cells, they combine to form tissues. So cells performing similar functions, these are known as tissues. Okay, then tissues combine to form organ, and then organs combine to form organ system. So a particular function is carried out by a group of cells at a definite place in the body. Similarly, different functions are carried out by different group of cells in an organism. Okay, so they are saying basically different tissues are performing different kind of functions. And what is tissues? Tissues are the cells, okay, which are grouped together, and these cells are performing similar functions. So there is division of labor in the multicellular cells. So you can give examples of liver cells, okay, heart cells, cardiac cells, all right. Then nerves, neurons, nerve cells, okay, blood cells. Next is cell is a basic unit of life. You have to discuss this statement briefly. Means in short, why is cell called the basic unit of life? So let's have a look at the answer. See, cells are the basic unit of life, and cell can perform all the biochemical processes, okay, which are required to live. Okay, you can see in prokaryotic organisms, single cell is performing all the function, and all the organisms, living organisms, they are made up of cells. So cells can perform the biochemical processes that are required to live. Now, the basic need for the survival of all the organisms are actually same. All living organisms need to respire. By respiration, energy is generated in the form of ATP. Then all the organisms, they need to digest the food to obtain energy. And by all of these metabolic reactions, waste is generated. Waste is generated in the form of carbon dioxide. Waste is generated in the form of nitrogenous waste. So this waste has to be removed out of the body. Otherwise, it will be toxic. So all of these processes are done by the living organisms. Now, cells are capable of performing all these metabolic functions of the body. Means cells are able to perform all the life processes. That's why cells are known as the basic unit of the life. So they are the structural unit of life also, and they are the functional unit of life as well. Means organisms are made up of cells, and the cell is performing all the functions, okay, required for the survival, required to live. Next question: what are nuclear pores? And you have to state their function. Okay, what is the function of nuclear pores? So let's have a look at the answer. See, nuclear pores, they are the tiny holes present in the nuclear membrane of the nucleus. So nucleus is surrounded by the nuclear membrane, also known as the nuclear envelope. This nuclear envelope consists of this nuclear pores. Now, these nuclear pores, they are formed by the fusion of the two nuclear membranes. The nucleus is also surrounded by two membranes. Okay, so these nuclear pores, they are formed by the fusion of the two nuclear membranes. Okay, means the nuclear membrane is absent also, you can say here. So these holes allow specific substances to be transferred into the cell and out of the cells. Okay, again there is a selective transport here. Like messenger RNA, after transcription, messenger RNA is produced in the nucleus. And this messenger RNA, it is transported to the cytoplasm through these nuclear pores. Some of the proteins also, they enter into the nucleus, and some of the proteins, they you can say, leave the nucleus as well through these nuclear pores. Okay, so these pores allow molecules such as RNA and proteins to move in both the directions. Okay, between the nucleus and the cytoplasm. So there is exchange of material between the nucleus and the cytoplasm. There is exchange of molecules between the nucleus and the cytoplasm. This is an image showing you nucleus. So within the nucleus, you have the nucleolus, where ribosomal RNA synthesis occurs. This is the nucleoplasm, which is the fluid of the nucleus. Within the nucleus, you have the chromatin material, which is made up of nucleosomes. Okay. Now, these are the nuclear pores through which these molecules, they are transported across the nucleus. Now, next question, both lysosomes and vacuoles are endomembrane structures, yet they differ in terms of their functions. Okay, what is the answer here? Now, lysosomes are membrane bound vesicular structures holding a variety of enzymes such as lipases, proteases, and carbohydrates. The purpose of lysosome is to digest the worn out cells. So, lysosomes are also known as the suicidal bags. They consist of the digestive enzymes. Okay, they are involved in intracellular digestion of foreign food particles and microbes as well. Sometimes they act as suicidal bags also. So if the cell is damaged or if there is some problem with the cells, these lysosomes release the digestive enzymes and they sort of digest the cells because they have digestive enzymes. Okay, so they will digest all the biomolecules as well. They are involved in self-digestion of the cells. That's why they are known as suicidal bags as well. This is an image showing you lysosomes. Okay, so they have these hydrolytic enzymes in them. Okay, they are surrounded by a this single lipid layer as well. Then you have these vacuoles. Vacuole is surrounded by a membrane known as the tonoplast. Okay, so vacuoles also can store certain molecules in them. There are different kind of vacuoles as well, like gas vacuoles as well. Now, vacuoles, they are kind of waste disposal system of the okay, these lysosomes are a kind of waste disposal system of these cells. On the other hand, vacuoles are storage sacs found in the cells. They might store the waste products also of the cell because these waste product can be toxic to the organelles. Okay, they can be toxic to the components of the cell. So these waste material, they are shifted into the cytoplasm, and tonoplast help in this. So tonoplast help in the active uptake of the molecules within the vacuoles. In unicellular organisms, food vacuoles contain the consumed food particles. Okay, so like in, you can say, in amoeba also food vacuoles are formed. So they engulf the food and they store this food in the food vacuoles. They also play a role in expelling the excessive water and some waste from the cells. these vacuoles also help in the osmoregulation as well. Now, next one, describe the structure of the following with the help of labeled diagrams. Okay, nucleus and centrosome. So let's have a look at the answer. Let's talk about the nucleus first. See, nucleus controls all the cellular activities of the cell. So it is basically the control center of the cell. Like your brain and spinal cord, these are controlling all the activities of the body. Here, the nucleus is controlling all the cellular activities. Okay, activities going within the cell. Nucleus, this is spherical in shape. Here you can see this is the nucleus. It has outer and an inner membrane. This is the nuclear envelope. These are the nuclear pores. Okay. Here the nuclear pore complex is formed. Okay. Within the nucleus, the chromatin is present, DNA is present in condensed form. Okay. Then this is endoplasmic reticulum, which is associated with the nucleus. Okay. So the genetic material is present within the nucleus. Okay. Now, nucleus is composed of nuclear membrane, also known as the nuclear envelope. So it is double membrane separating the contents of the nucleus from the cytoplasm. Now, space between the outer nuclear envelope and inner nuclear envelope is known as the perinuclear space. Okay. Here you can see in this image, this is the perinuclear space between the outer membrane and the inner membrane. Okay. So nuclear membrane has tiny holes known as the nuclear pores. These pores or these holes allow specific substances to be transferred into a cell and out from it. Okay, what molecules we have discussed, such as RNA and certain proteins. Next is nucleoplasm, also known as the nuclear matrix. So, nucleoplasm, this is the homogeneous granular fluid present inside the nucleus. Now, it consists of nucleolus and the chromatin. So, chromatin is what? Chromatin consists of the DNA along with the histone proteins and some non-histone proteins. So, chromatin is made up of DNA, histone proteins, and non-histone proteins. Okay, nucleolus, this is a region without any membrane. In nucleolus, ribosomal RNA synthesis occurs. Okay, nucleolus is a spherical structure that has that is not bound by any membrane. Okay, you can see this is the nucleolus. This is not bound by any membrane. Okay, this is the nucleoplasm you can see. These are the chromatin material, which is DNA in very condensed form, because DNA is wrapped around the histone forming a nucleosome. These nucleosomes are attached together to form the chromatin. Okay, when you observe chromatin under electron micrograph, it will you will observe like beads on a string structure. These beads are nothing but nucleosomes. Each nucleosome carry 200 base pair of the DNA. Okay, so this nucleoplasm is rich in protein and RNA molecules and is the site of ribosome formation. So this nucleolus, you can say, nucleolus is rich in protein and RNA molecules and it is the site of ribosome formation. Ribosomal RNA synthesis also occurs here. Chromatin, as you know, this is entangled mass of thread-like structures. It contains DNA and some basic proteins called as histones. Along with this, some non-histone proteins are also present. Let's talk about the centrosome. So centrosome consists of two cylindrical structures called the centrioles. Okay. So each centrosome consists of two centrioles. The centrosome is divided during the cell division. So these centrioles which are present within the centrosome, they lie perpendicular to each other. Okay, centrioles, they will lie perpendicular, means 90° to each other. Now they have cartwheel-like organization. Okay, this is the centrosome. You can see they consist of these two centrioles, and these centrioles, they are arranged perpendicular to each other. You can see 90° to each other. The centrioles help in the spindle fiber formation. Okay, clear? And this is the axoneme. You can see this axoneme is present in the cilia and flagella actually, which have 9+2 arrangement. Okay, doublets of microtubules on the periphery, and in the center you have two microtubules. So this is 9+2 arrangement of the axoneme, which is present in the cilia and flagella. Okay, but here we are talking about the centrosome. Okay, they have cartwheel-like organization. Okay, so centriole is made up of microtubule triplets that are evenly spaced in a ring. Okay, so this is what is cartwheel-like arrangement. In cartwheel-like arrangement, what is happening? There are triplet of the microtubule. Okay, and these triplets, they are also joined together. Adjacent triplets, they are joined together with each other. Okay, they are 9 microtubule triplets actually. Okay, microtubules, they are made up of tubulin proteins. Okay, adjacent triplets, they are linked together. These microtubule triplets, they are linked together in the cartwheel-like arrangement. Now, there is a proteinaceous hub in the central part of the centriole. Okay. Now, the proteinaceous hub, which is in the center, is connected to the microtubule triplet via radial spokes. These centrioles help in organizing the spindle fibers and astral rays during the cell division. Okay. So, from these centrioles will arise the microtubules, and it will help in the organization of the spindle fibers. Okay, these spindle fibers are very important for the separation of the chromosomes. Okay, now they also form the basal body of the cilia and flagella as well. Now, next question. Okay, what is centromere? And how does the position of the centromere form the basis of classification of chromosome? And you have to support your answer with a diagram showing the position of centromere on different type of the chromosomes. Okay. So let's have a look at the answer. See, centromere, this is the primary constriction present on the chromosomes. Okay. And centromere consists of kinetochores. Okay. And on these kinetochores, the spindle fibers, they attach. Clear? And the two chromatids, they are held together at the centromere. Okay. Now, chromosomes are divided into two types based on the position of the centromere. Okay, there are 4 types of the chromosome. First is metacentric chromosome. Okay, so these chromosomes, in metacentric chromosome, what is happening? The centromere is present in the middle and divide the chromosome into two equal arms. Okay, the two arms, P and Q arm, that will be equal because the centromere is in the center. Here, this is an image showing you metacentric chromosome. Centromere is in the center. These are the two chromatids you can see. These are the sister chromatids. Okay, this is the centromere, which is the primary constriction. Two chromatids, they are held together at the centromere. This is the P arm, this is the Q arm. Both the arms are of equal length. The next one is submetacentric chromosome. Here the centromere is slightly away from the middle and is known as submetacentric chromosome. Here one arm will be slightly longer than the other arm. Okay. So here you can see P arm is shorter as compared to the Q arm because centromere is slightly away from the center. Now next one is acrocentric chromosome. Now here what is happening, the centromere is located close to one of the terminal end. Okay, so still centromere is not at the terminal end, it is close to one of the terminal end, and this type of chromosome is known as acrocentric chromosome. In this, one arm will be extremely long as compared to the other arm. So you can see this P arm, this is extremely short as compared to the Q arm. Okay, here centromere is close to one of the terminal ends. Okay, and here you can see this is the chromatin in the chromosome. Then the last one, fourth type of chromosome, telocentric chromosome. Actually chromosome, the three main type are that we have discussed. Telocentric is not that common, okay? But this type of chromosomes also exist. Okay, metacentric, submetacentric, and acrocentric. These are the three main type, and this fourth type, this also exists, but those three were the main type. Now, the chromosome in which the centromere is located at one of the terminal end, this is known as telocentric chromosome. So here the centromere is at the terminal end. Okay. So here you can see these are telocentric chromosome. Here the centromere as the terminal. And the shape of the chromosome is like, you can see small I. These are the chromosome arms here. Okay, these are telocentric chromosomes. All right. So with this, we have discussed the NCERT exercises also. Okay. So we have discussed all the concepts as well. Clear? So with this, we will end this session. I hope this session was quite knowledgeable for you. Now we'll meet you in the next session. Thank you very much for listening.